transitions like the timing of reproduction, the timing of migration, and other sorts of issues. What I'm going to try to convince you of tonight is that we also need to take evolutionary dynamics into account when we're considering how natural populations and species are responding to climate change. All landscapes are heterogeneous to a certain extent, and many species have evolved to be locally adapted in response to divergent selection that they experience across their landscape. So there may be populations adapted to shorter growing seasons in higher elevation locales and cooler and warmer, more arid, low elevation conditions. Local adaptation is a process that has occurred repeatedly across a huge diversity of organisms, ranging from vertebrates to invertebrates and plants. Typically under local adaptation, what we see is that genotypes that evolved in their home environment have elevated fitness relative to genotypes or families from other locations. So you often see what we call crossing reaction norms. So here is just a schematic of what this looks like, where you have fitness on the y-axis and then the environment where you're assaying fitness on the x-axis. Genotype 1 evolved in environment 1. This could be a maternal family or a clone or some sort of line, and it has elevated fitness in its home site and depressed fitness in the contrasting site. And you see the same pattern for genotype 2, which evolved in environment 2. I do hope that you remember, though, back to basic geometry class that you probably had in high school, where you were told not to connect 2 points with a straight line, because it's not actually great. And there's a huge amount of environmental variation that can occur across the range of species. In addition, local adaptation studies have done an excellent job at categorizing patterns of local adaptation, principally through these reciprocal transplant studies, but we know very little about the process of local adaptation. So there are a number of agents selected agents that can contribute to local adaptation. And these are just a handful of them. Photoperiod, temperature, the level of drought stress. In high elevation and high latitude systems, snow dynamics can be a major factor in the extent of local adaptation. Edaphic factors, so those are soil characteristics. And then you'll notice that I ran out of space, so we had to put the biotic community way down at the bottom, but there are a huge number of biotic interactions, including mutualistic interactions as well as antagonistic interactions that can lead to the evolution of local adaptation. In most landscapes, species are distributed across complex environmental gradients, where conditions change gradually across the range of the species. So in most cases, you don't see two distinct environments that a species can encounter. Rather, species have these broader distributions. So the question then is, how will climate change influence patterns of local adaptation along gradients? And I'm going to argue that evolutionary biologists have not done a good job at categorizing or cataloging local adaptation to environmental gradients. But we can take a page out of the book from forestry. So foresters have been doing a really superb job at trying to understand how tree species are adapted to broad environmental gradients that they experience across their ranges. And these studies originated as mechanisms for identifying seeds to plant into reforestation efforts across the range of species. These trials are principally called Provenance trials, and they have two primary advantages over the traditional reciprocal transplant experiments that evolutionary ecologists have come to love. So the first advantage is that, unlike evolutionary ecologists, foresters in these provenance trials sample populations from across the range that have evolved in response to really disparate environmental conditions that they experience. So this is an example from a provenance trial of lodgepole pine that was started in British Columbia in the 1970s. And up in this map, so here's Washington State and then British Columbia, you can see the dots represent all of the various accessions or seed collections that foresters have used. And then they throw all of these seed collections into numerous common gardens planted across the range. So they're interrogating fitness and traits in many, many different environments, natural environments, in which this species originally evolved. And they ask, how much does the origin site of populations or seeds influence their fitness? So in these provenance trials, you often see patterns like this. So on the y-axis, we're looking at height of these plants at 20 years after planting. You could use any fitness component that's relevant to the study that you're interested in. Here they're using growth of these trees because it's very hard to assess fecundity in long-lived trees over relatively short human timeframes. On the x-axis, they're looking at the mean annual temperature of the source population for each of these accessions. So each of the data points represents a single accession or seed collection that they planted into this one particular common garden called COM, and they evaluated the height. And you often see patterns like this. This is a negative quadratic curve where the peak fitness is seen in plants that evolved in a similar environment as this garden. So on the x-axis, I've starred the mean annual temperature at this particular garden. And you can see that plants that evolved in cooler environments are shorter. Plants that evolved in warmer environments are also shorter. So you see optimal performance of seeds that evolved in environments similar to this common garden. One of the reasons that it's really critical to evaluate local adaptation in a changing climate is that climate change has dramatic consequences for many of these agents of selection that promote local adaptation. Many, but not all. And what's really interesting about this is that climate change is decoupling these historically correlated environmental conditions that natural populations have evolved in response to. So climate change isn't changing photoperiods, it's not changing the length of the day, and it's not changing the amount of light that organisms are experiencing. But we all know that climate change has pronounced consequences for temperature, with increasing temperatures globally. Temperatures have increased by 1.2 degrees Celsius or so since the 1880s and are projected to increase dramatically in the future. These increasing temperatures and altered precipitation regimes can lead to increased drought stress. In high elevation and high latitude locations, we're seeing reduced snowpack, which can have very profound consequences for native species during the growing season. And we also see really pronounced changes to the biotic community. So climate change is decoupling these agents of selection and could in fact be disrupting long-standing evolutionary patterns of local adaptation that have taken many, many hundreds of generations to evolve. So the questions that I'll be getting at in this talk involve the evolutionary consequences of climate change. We're really interested in how climate change imposes new patterns of natural selection on natural populations and how those populations can evolve in response to this new selection. Do they have sufficient genetic variation to evolve? And we do this over elevational gradients, because you can capture a huge amount of climatic variation in very short spatial scales. So this is just a schematic of the mean annual temperature across elevations in our study region and mean annual precipitation. And essentially what we see is hot, arid, low elevation locations with very long growing season, transitioning into cooler, moister, high elevation locations with a very, very short window of opportunity for species to flower and fruit and complete their life cycles during the growing season. So the main question that we'll be talking about today is, will climate change disrupt these long-standing evolutionary patterns? And to do that, first we need to evaluate local adaptation to elevation in contemporary environments so that we get a sense of how well adapted these local populations are to the environments that they experience. We're then going to look at phenological responses to climate change, principally through the timing of reproduction. So how has climate change already altered when these plants reproduce? And that will provide a really nice segue into understanding some of the evolutionary consequences of climate change. The focal species that I'm talking about is this beautiful alpine mustard called Bucera stricta. It's common in undisturbed habitats throughout the western United States, where it grows in the US Rocky Mountains from Utah all the way up through the Canadian Rocky Mountains and into Alaska. So it has this incredibly broad elevational gradient and latitudinal range. And it has, there are biogeographic studies that show that the species has expanded and contracted its range in response to the Pleistocene glaciers. So as the glaciers formed, the range contracted. As the glaciers retreated, the range expanded. So we know that in a very, very short geological time frame, the species can alter its distribution patterns. And the question is, now that climate change is extremely rapid, how will this rapid environmental change influence the biology of this native species? It's a short-lived perennial. It's primarily selfing, so it doesn't have a lot of interactions with pollinators. It has the most boring mechanism of seed dispersal that I've ever worked on. So basically makes siliques that dehisce, they open up and the seeds fall right underneath mom. It's an extremely experimentally tractable system, so we can plant thousands of plants in the field and look at their fitness and traits in many different locations. And it does have a fully sequenced genome. I won't be getting into that today, but it's a benefit of working in this system. My primary field site is in kind of south central Colorado at the Rocky Mountain Biological Lab, which was established in 1928 in an old mining area. And I'd like to acknowledge that this is the unceded sovereign territory of the Ute people, who prior to the US acquisition of Colorado had a range that expanded. They lived in essentially all of Colorado and much of Utah. When the US acquired this land after interactions with Mexico in 1848, gold was discovered in Denver about 10 years after that initial acquisition, and the Ute people were consequently forced off their land through a series of treaties that the US did not abide by. So this is just a change in the Ute-dominated landscape over time, and there are now about 3,000. The tribal membership of the Ute people is about 3,000. They mostly live in a reservation north of Salt Lake City in Utah. And if you'd like to learn more about the Ute people and their continued struggles with the US government for their sovereign territory, there's a great website that they manage called utetribe.com. And if you would like to learn about the native peoples of Georgia or other locations, there's this wonderful Canadian website where you can go to find more information about people who lived in the places that you love. So in this location, we've done a very large scale common garden experiment at 5 gardens that vary in elevation from 2,540 meters all the way up to 3,300 meters. And I'm not good at translating from meters into feet in my head, but this range is several thousand feet in elevation difference. And over the course of the past decade, we've planted somewhere in the neighborhood of 180,000 seeds and seedlings into these gardens to evaluate their fitness. We've been taking a provenance trial approach, so we use seeds that we've collected from across this range to evaluate clinal variation in local adaptation to elevation. And for the first part of the data today, I'll be focusing on 2 gardens exclusively, though later in the talk I'll show you data integrated across all of the gardens. But these 2 gardens are good representatives of what's happening across this range. So these 2 gardens, just to kind of orient you to what you'll be seeing in the next couple of figures. First, on the y-axis, there'll be some metric of fitness, some metric of performance. And those metrics that you'll see will be germination success, survival, and fecundity. So these are really important performance metrics for this plant. And on the x-axis, you'll see the elevation of origin of the families that we're including in these gardens. And based on the provenance experiments, we can make really clear and distinct predictions about how local adaptation should look, what these figures should look like. So what you'll see on the x-axis are these stars indicating the elevation of the garden. And in all cases, we expect these negative quadratic curves where fitness or performance peaks in families that evolved at similar elevations as the garden where we're doing this work. Now, I want you to keep in mind that these are not families that we collected exclusively from those gardens. We collected families from across this range. So if a family evolved at an elevation similar to one of these gardens, we would expect it to have high fitness, and we would expect low fitness in lower and higher elevation accessions. So we do see clear evidence for local adaptation and germination. This is one of the seed grids that we put out, and a little Bucera seedling germinating. All of the data points that I'll show you today represent family level averages. So we plant many individuals, siblings per family, then we average them to get a good representation of what the family is doing. So at this lower elevation location, we see that we see this clear pattern of a decline in germination success with increasing elevation, and that's consistent with our expectations. And we also see this negative quadratic curve in this higher elevation location, where genotypes from an elevation that's very similar to this garden have high fitness relative to those from lower or higher ends of the spectrum. I do want to point out that this is the last time that you'll see evidence for local adaptation at this higher elevation location, and it's really important. My plug here is that it's really important for scientists to evaluate fitness components at many different life history stages across the plant. The seed to seedling transition, this germination stage, is often overlooked in evolutionary ecology studies because it's challenging to deal with small seedlings, but it's really crucial to get those data. We see similar patterns in survival for this lower elevation garden, where the odds of survival decline by about 10% for every 100-meter increase in source elevation. But there's some evidence for maladaptation in the higher elevation location, where that negative fitness curve, that negative quadratic curve, is shifted down from the elevation of the garden. Finally, for fecundity, you're probably bored of seeing this pattern, but we see the same pattern at this low elevation site, where the fecundity of the plants declines with the elevation of origin, again, consistent with local adaptation, but there was no pattern, we detected no pattern in the higher elevation location. So far I've been showing you fitness-related patterns, but these plants are complex organisms that have a variety of different traits that have likely evolved in response to divergent selection that they experience across their range. So we can turn our attention now to looking at some of the traits of that these individuals express. And one trait is flowering time. Flowering time is probably the best characterized trait in biology, in plant biology. It's very well characterized at the developmental level, the molecular level, the genetic level, and even the ecological level. And here we see patterns that are very consistent with expectations, where there are these clear climbs in flowering phenology. Plants that evolved in high elevation locations flower very rapidly. And that makes sense because these plants have a very, very short window to reproduce during the growing season. Snow melts off in July and it comes back on in September. So they need to get things going. In contrast, plants from lower elevation gardens can be they can take a little bit more time to flower. And in doing so, they're likely devoting resources to other functions like protection against insect herbivores. But you can see that these patterns are very similar for both of the gardens. We also see these genetically based declines in size at flowering. So high elevation genotypes flower rapidly, they flower at a small stature, and they have a much shorter duration of flowering. Like I said, this is likely a response to long-term selection in those high elevation locations for very rapid development. We can also look at physiological parameters. So here we basically ground up leaves and we assayed delta C13, which is a stable carbon isotope in those leaves, which is a very, very good proxy for how efficiently plants are using their water. Here we have an aridity. I've coded each of these populations by the aridity index of the population of origin, where red colors are very arid, and these bluer colors are much less arid, they're more mesic. So not surprisingly, we see that low elevation plants are very water use efficient. it's an arid location, so this high water use efficiency is likely a response to the aridity in that location. And the higher elevation plants are a little bit more profligate in their water use, and that might be because they need to use water more rapidly to complete their rapid development. What's interesting about water use efficiency is it's extremely context dependent. So we studied the same exact plants. These are still family level data, but the underlying individuals that go into these data points are the same exact individuals across 3 years. One in 2012, which was extremely dry, where snow melted 30 days earlier than average. 2013 was dry with an early snow melt of about 10 days. And then 2014 was incredibly benign. And what we see is that we can pick up these clear genetically based declines in water use efficiency under drought stress, but they disappear under benign conditions. And this again is a plug for doing these studies in ecologically relevant ways. If we only modeled benign conditions in the greenhouse, we wouldn't be able to detect this clear context dependency. And I say that because I know that. So my student, Rachel McTavish, who's finishing her PhD soon, did a really lovely multi-generational study in the greenhouse where she exposed plants to drought stress, and then she also exposed them to nutrient stress. Here, I'm just going to be focusing on some of the results from her work, which she did in collaboration with Sunny, an undergrad in the lab. And I'm just showing the results that are consistent or fit within the context of the story that I'm telling you right now. She has a huge amount of other really interesting data about trade-offs that these plants experience across their life cycles. In her well-watered environment, we don't see this clear suggestion that low elevation plants have elevated fitness. And in fact, in well-watered conditions, there's a slight indication that performance or fecundity increases with the elevation of origin of the plants that she used. But when she exposed plants to moderate drought or even more severe drought, she saw clear evidence that these low elevation drought adapted plants have elevated fitness. So as I mentioned earlier, it's very challenging to get at the agents of selection that influence local adaptation. And really there have only been a handful of studies that have addressed these agents, have identified the agents of selection that influence local adaptation. And one thing that's great in this system is that we compare ecologically relevant work in the field with these greenhouse experiments. One thing that's also exciting is that selection favors local trait values. So what I'm about to show you are 3 figures on natural selection. So on the y-axis, you'll see some metric of fitness. It'll be either flowering success or fecundity. And then on the x-axis, what you're going to see is the traits that we looked at. And I did this work in collaboration with Susanna Vahimar, a former postdoc in the lab, who's moved on to Davidson College. And the cool thing here is that on the x-axis, we've highlighted the local trait values. So these are the trait values that are expressed by local plants, plants that originated in an environment similar to the garden. And in all of these cases, we see these negative quadratic curves, which are consistent with stabilizing selection, where the peak fitness occurs more or less exactly where the local populations sit. And that's super exciting because there aren't that many studies that actually document that natural selection favors local trait values in the field. And this suggests that these genetically based declines that I've been showing you are adaptive responses to long-term selection that varies across the landscape. Now, I mentioned earlier that bluegrass has relatively boring interactions with other species. It's self-pollinating, has boring seed dispersal mechanisms. But the great thing about Bucra stricta is it does have many interactions with herbivores. So this is a caterpillar consuming a leaf of one of our experimental plants that's already started to flower. So we can look at the extent to which there may be local adaptation in the plant responses to herbivory. So here, what we'd be doing, these are predictions of the extent of leaf damage. So every year I go out into the field and count thousands of leaves to assay the amount of damage that they're receiving from herbivores as a function of the elevation of origin. And in this case, we predict positive quadratic curves, where the least amount of damage should occur in accessions that evolved in elevations similar to the garden. We don't, in fact, see this patterns. So if there were local adaptation, we would have expected those curves. Now, at our lower elevation garden, we do see that the extent of herbivory increases with source elevation. But we see that same exact pattern at the higher elevation garden. And I often don't believe patterns until I've seen them repeatedly. This is the same pattern I've seen across all 5 of these experimental gardens from 2013 to the present. We always see these genetic declines in herbivore resistance such that low elevation populations are resistant to herbivores and high elevation populations are not. In fact, there's a genetic trade-off between rapid development and defense against herbivores. So those high elevation plants that need to get things done quickly don't have resources to devote to anti-herbivore defenses. Whereas those lower elevation plants that can take their time in developing can devote the resources that they need to a variety of different anti-herbivore defenses. And you might be thinking, how is this relevant to climate change? Herbivores may have a much more rapid response to climate change than plants. This could occur because herbivores are highly mobile, so they could move up mountains quickly, and or because herbivores have fast generation times, so they could expand their populations in situ much faster than the plants can. That could put high elevation plants at risk of decline when they're exposed to novel herbivore communities that they had never experienced throughout their evolutionary history. So to evaluate plant-arthropod herbivore interactions under climate change, I teamed up with Elena Hammond, who's a postdoc in the lab, as well as Cam Blevins, who's an undergrad and a grad student in Nam Jamil, and Steve Franks, who's a professor at Fordham. We did a phylogenetically corrected analysis trying to understand how these interactions could shift under climate change. And what's really cool about this is that there are nice, clear mechanisms that we can point to with these patterns. So a meta-analysis essentially allows us to go into the literature, pull out papers that are relevant, and evaluate plant and herbivore responses to various treatments that reflect climate change. So I'm going to show you schematics of predictions first, and then I'll show you the real data. We, for the herbivore meta-analysis, we looked at a variety of different treatments that were associated with climate change, including increased carbon dioxide concentration, increased drought, increased amount of fertilizer, and increased temperature. And then we also were able to look at different herbivore traits, including food consumption, development, growth rate, and a variety of other traits. So any values that fall to the left-hand side of 0 here indicate that climatic manipulations reduce herbivore traits, whereas any values that fall above 0 indicate that climatic manipulations increase this trait. So here are the results from this, and I'm going to just highlight the salient points. So first of all of the treatments that we were able to assay in these 62 studies of 57 arthropods show that arthropods are increasing their consumption rates in response to elevated CO2, drought, fertilizer, and temperature. This makes a lot of sense. As CO2 concentrations increase in the atmosphere, this fundamentally alters the leaf chemistry in plants, actually all tissue chemistry. You end up seeing greater rates of carbon to nitrogen ratios increase. And what that means is that herbivores have to consume more tissue to be able to get the nutrients that they need to complete their life cycles. So all of these metrics of consumption, all of these climate change factors influence consumption. We also see an increase in development time under increased CO2. So that means that the herbivores are developing slower. What's interesting is that increased temperature also influences development time, but it actually reduces development time. So insects develop faster under increased temperature, and I don't think this would surprise anyone. Arthropods are ectotherms. No one would be surprised that under increased temperature, they're developing more rapidly. So climate change increases food consumption across all of the factors that we were able to assay in this meta-analysis, and alters developmental timing. You might wonder what the consequences of these changes are for plants. We also wondered, so we did a complementary meta-analysis where we looked at CO2 concentrations, drought, and temperature in annual and perennial plants across a variety of tissue types. And just like the herbivore analysis, if the value is negative, it means climatic manipulations reduce damage from herbivores. If the values are positive, it means climatic manipulations increase damage. And what we found was that these climatic manipulations increased foliar damage in annual plants and floral damage in perennial plants. And that's really important because flowers are tightly linked to fitness. Clearly more research is needed at this point, but climate change increases both foliar and floral damage that plants receive from herbivores. Okay, so I hope in this first part of the talk that I've convinced you that there is clinal variation in fitness and traits, and this is consistent with local adaptation to climate change, or sorry, local adaptation to climate. And what I want to transition into talking about now is how plants are responding phenologically to climate change, and that will lead us to understanding the evolutionary consequences of climate change. All organisms undergo a sequence of life history stages, and they have to time the transitions between these stages really precisely to capitalize on favorable conditions and to reduce mortality going forward. So this is the life cycle of Bucera stricta, from seed germination in the spring to overwintering, flowering and subsequent growing seasons, producing seeds and dispersing them, and then overwintering again. You could imagine that there may be an optimal timing of flowering, because individuals that flower too early in the season may get hit by things like late season snowfall events or late season frosts or other late season winter events. And plants that flower too late in the season may not have sufficient time to complete their reproductive cycles prior to the onset of inclement conditions. So there may be this intermediate optimal flowering time where plants flower when their pollinators are available, and where plants within a species are all flowering at the same time to optimize the amount of outcrossing. So we can make a prediction similar to what we saw earlier with stabilizing selection operating on traits. We may predict that stabilizing selection should operate on flowering time. So here we're looking at fitness as a function of the timing of year of first flowering. And we predict that plants that flower too early in the season may have reduced fitness because they're too small or they risk frost damage. Plants that flower too late in the season have reduced time for reproduction, and they risk seed dispersal and development during adverse conditions. So we might expect there to be some intermediate optimal flowering time. This is not the pattern that we see, and this is something that's confounded me for probably a decade. So what I'm showing you right now comes from a much earlier study that I did when I was a postdoc, where we looked at selection in Colorado and Montana on the timing of year of first flowering, and we saw clear evidence for directional selection for earlier flowering. So plants that flowered early had elevated fitness relative to those that flowered later. In our more recent work in Colorado, we see the same pattern of directional selection for earlier flowering in basically all of the locations. Here I'm showing low elevation and high elevation, the gardens that I've been focusing on earlier. But what I want to draw your attention to is how much of a difference there is between the flowering time of the local plants in these locations. So these are the local accessions. Are here in the middle. These local genotypes are flowering 10 to 15 days later than the optimal timing of flowering. And this suggests that climate change may already be disrupting longstanding selection patterns. This is a pattern that others have recognized in the literature too. So this wasn't mine, but this is an earlier study from 2011 saying that a meta-analysis of phenotypic selection on flowering phenology suggests that early flowering plants are favored. That's exactly the pattern that we just found. So the question is why? What explains this consistent pattern when it's not what we would have expected? And one potential answer is climate change has already changed the conditions that plants experience. Climate change We know a lot, as I mentioned before, about flowering phenology and the cues that plants use to trigger flowering. And those cues are also cues that are changing with climate change. So there are some major flowering time pathways in model organisms, including temperature pathways. We also know that drought stress and snow dynamics can influence, can elicit flowering in a variety of different environments and species. And climate change has already shifted many of these climatic factors. So this is a heuristic model because I do not, in fact, have a time machine that would allow me to establish all of my experimental gardens in pre-industrial climates. If anyone finds a way to do that, I would really, really love to do it. My other academic pipe dream is that there's a control Earth that scientists can go and study so we don't have to worry too much about climate change. So heuristically, we predict that in pre-industrial climates, there should have been stabilizing selection operating on flowering time. But under contemporary climates, essentially climate change is pulling the rug out from underneath these locally adapted populations, shifting this fitness curve. So what we see now is a reflection of this shifted curve. And what we want to know is what might happen in the future. So many years ago, David Inouye, who was a professor at the University of Maryland and who's worked at the Rocky Mountain Biological Lab, came to me and said, "Hey, Jill, I have some data on Bucra. Do you want it?" And I said, "Please." So these are David's data looking at temporal trends in flowering phenology in Bucra stricta. And what I want to draw your attention to is that flowering time in this species has been advancing by 3.4 days per decade since 1973. And that number is highly consistent not only with phenological advancements in other species at the Rocky Mountain Biological Lab, but it's also highly consistent with phenological advancements in a diversity of organisms there, as well as plants, butterflies, amphibians in many different locations in the world. We can try to understand what are the factors that cause these shifts by looking at correlates of phenology. So here we're looking at the timing of your first flowering as a function of spring temperature, and you see a clear pattern where in years with delayed flowering, the temperatures were cooler. In years with accelerated flowering, the temperatures were warmer. But temperature in this system is likely not the primary driver. The likely primary driver is snow. Snow dynamics are incredibly important in shaping the biology of high elevation alpine ecosystems. And that snow insulates plants from frost damage that they would experience otherwise. And it also snowmelt provides most of the irrigation for this system throughout the growing season. Climate change and warming winter temperatures have reduced the amount of snowpack in the winter. It's actually shifting precipitation from snow to more rain. So the amount of precipitation hasn't changed, but you're not seeing the buildup of these huge snowbanks that we did in the past. It's also advancing the timing of snowmelt in the spring. So these are observed and projected changes in snow dynamics where we're already seeing advancements in the snowmelt timing in spring, and there may in fact soon be delays in snowfall in the autumn. So essentially warming winter temperatures reduce snowpack, and warming spring temperatures advance the timing of snowmelt. So we can go back to David's data and correlate flowering phenology with the timing of snowmelt, and lo and behold, we see a nice pattern. But as you know, this is a correlation and it's not causal. The other thing to keep in mind is that this is not a one-to-one pattern. The slope of this line is about a third. So for every day change in snowmelt, there's about a third of a day of change in flowering time. And that suggests that snowmelt is not the only factor that is influencing flowering time. So I teamed up with a graduate student who has long since finished his PhD, Zach Giesen, who is doing a variety of snow removal experiments for his own work. And he came to me and said, "Jill, I have some Bucra data. Do you want it?" And I said, "Yes, please." So this is mostly just to show you that Zach's experiment was effective at changing the timing of snowmelt. So this is just control versus snow removal treatments. And his experimental treatment, so this is Zach standing in one of the plots that he's shoveled out, he induced a 17.4 day advancement in the timing of snowmelt. So what's really cool now is that we can go back to David's data And we can say for a 17-day increase in the timing of snowmelt, what is the change that we would predict in the timing of flowering? And it turns out that that change is about 5 to 10 days. So these are the flowering time data from for Bucra stricta from Zach's plots. Again, you'll remember we predicted a 5 to 10 day change in the timing of flowering. And in fact, what he found was a 7-day change in the timing of flowering. So what's exciting about this is that it shows that we can use this relatively simple experimental manipulation to simulate long-term trends in climate change in this region. And that can give us a much better predictive tool for understanding how populations may respond to climate change going forward. So as I said before, we have this heuristic model, and the question is, under future climates, will local populations fall so far from these fitness peaks, or could they potentially maintain sufficient genetic variation to adapt to ongoing changes? In my lab, we really study how climate change induces local maladaptation, imposes novel patterns of selection, the extent to which phenotypic plasticity can contribute to biological responses to climate change. And then we also address whether genetic constraints or limited genetic variation could inhibit adaptive responses to climate change. And we ask the extent to which gene flow may play a role. So what I'm going to focus on for the next couple of minutes is just maladaptation. So we talked before about these fitness predictions that we could draw from the Provenance trial approach, where you would expect these negative quadratic curves. We also make a prediction that under climate change, you see a reduction in fitness across all genotypes from everywhere, because climate change has such stressful responses for natural populations. And as a first step in addressing this second prediction, I did another phylogenetically corrected meta-analysis where I went into the literature and pulled studies that were that analyzed some metric of plant fitness in natural environments in the field in response to climatic manipulations. So in these next two slides, any value that's over 0 suggests that climatic manipulations or climate change increases fitness, and anything that's under 0 suggests that climate change reduces fitness. I did 2 different fitness components. So the first was fecundity. Infrared heating lamps depress fecundity. Infrared heating lamps are a very good but extremely expensive mechanism for simulating climate change, because they increase temperature and draw down soil moisture, but they're very expensive to maintain. We also looked at binary components of fitness. So these are things like germination success and survival, and found that multiple manipulations depress germination, establishment, and survival of plants generally. And the reason that this matters is that reductions in fitness within a population can depress the adaptive population of those populations. Or the adaptive potential of those populations. They just may no longer have the amount of genetic variation that's necessary for an adaptive response to climate change. And what's an interesting and novel point is that reductions in fitness can also alter the migratory potential. So if fewer seeds are being produced, then fewer seeds are capable of migrating to new locations at higher elevation locales or in higher latitude places. We can also make a third prediction, which is that under climate change, this peak in the fitness is actually going to be shifted to lower elevation locations. So as climate change proceeds, any given location will become more arid and hotter, and plant populations that evolved in arid and hot environments at low elevation locations may actually thrive in those new conditions. So what I didn't tell you at the beginning of the talk is that in this large-scale common garden experiment that we've been doing in the field, we're actually manipulating snowpack in all of these gardens. So this is one day in 2014, immediately before snow removal. And then this is 10 days later, where you can already see that the snow has melted out in these snow removal plots, but that our control plots still maintain snow. Early snow removal alters plant phenology, exposes plants to frost that they wouldn't otherwise experience, and decreases soil moisture. So the last couple of slides are going to integrate across all of the gardens. So here on the x-axis, we're looking at elevational difference. So essentially what I've done is I've taken the source elevation and I've subtracted the garden elevation. Perfect local adaptation would be a negative quadratic curve that peaks at 0 elevational difference. If that curve is shifted lower, it suggests that low elevation genotypes have a fitness advantage. If the shift is higher, it suggests high elevation genotypes are advantageous. So what we find is that there's clear evidence for local maladaptation under snow removal conditions. So this is our snow removal treatment. And there was a 70% reduction in the odds of reproduction for every 1 kilometer increase in elevation difference. So we see this clear pattern where low elevation genotypes are successful. So clear local maladaptation under snow removal. We can also map the control plot on here, and we see that there's a peak fitness at negative 277 meters in the control. And what that suggests is that there's even local maladaptation in the control. It's not as bad as what we see in the snow removal treatment, but it occurs even in the control. This was a pattern that kept on occurring, so I started to wonder whether we could recover local adaptation if we added snow to these sites. So we piled these bins on, we threw a lot of snow in them, and we assessed the germination success of seeds. So here you're going to be looking at snow removal control and snow addition plots, again, across all gardens and germination success. And in both snow removal and control environments, we see that low elevation genotypes are favored, but we are capable of recovering local adaptation in this snow addition treatment. We've actually replicated this experiment and found the same exact results. So local adaptation reemerges when we actually add snow, creating a climate that's more similar to historical climates. There are clear consequences of these shifts in fitness for population growth rates. So this looks at population growth rates across all 5 of the gardens, so from the low elevation to the highest elevation location. A population growth rate of 1 indicates that a population is stable. Anything below 1 means a population is contracting, and anything above 1 means the population is growing. So I modeled these population growth rates using a reciprocal transplant approach. So anything that's in these squares represents local genotypes. But the key point here is that within the context of this experiment, we're already seeing large-scale population contractions in the warm trailing edge of these populations. So to summarize what we've talked about, we've looked at clinal variation in fitness, life history, and ecophysiology, and all of these patterns suggest that there's local adaptation to historical climates. Maladaptation occurs in contemporary environments, but it also becomes more pronounced under snow removal. And climate change may have the greatest effect on early life history stages like the seed to seedling transition. So with that, I am happy to take any questions that you had, and I want to thank you for your time and attention this evening.
Wonderful, that was so great. I like the idea of having a controlled earth. I think that's needed. So we do have a couple of questions, and it looks like there might be some more questions rolling in too, but we have one from Kevin Burke asking, does the lodgepole pine study exemplify the NPS requirement that restoration plantings within national parks must be via seed collected from within the local ecozone or ecotype?
So I don't know if I can answer that question directly because I don't know the particulars of that regulation. But I do know that many of these provenance trials are occurring primarily within Canada and not as much within the US. And the whole entire point of those provenance trials was to identify seeds that they could use for reforestation elsewhere. And the other thing that they are starting to do with those data is to evaluate the extent to which populations are already experiencing climate change. So I don't have enough information about the national park system to be able to answer that question. Gotcha.
Okay. And then we have another question from Jennifer Boyd asking, hi Jill, I'm very interested in local adaptations within the context of climate change responses. One thing I've thought about in the context is the need perhaps to take local adaptations into account when developing and using like species distribution models to predict future species distributions. Can you comment on applications like this?
Yeah, I think it's a great idea. I think that local adaptation makes it a little bit more challenging to make solid predictions about range-wide dynamics if you don't account for local adaptation, because many of those species distribution models assume that all populations have the capacity to migrate and to thrive in new environments, but you may end up seeing many locally adapted populations that have very, very different biologies from each other. So I would like to actually start doing some species distribution modeling with Bucra in the context of local adaptation. I think the predominance of local adaptation, or the ubiquity of local adaptation, might make our predictions a little bit more grim. I think it may be harder for some locally adapted populations to expand past their current range, although it's possible that assisted gene flow could also enable populations to expand their ranges or thrive in new environments.
Great. And then we have a question from Stuart Whipple saying, even though you said that the dispersal of this species is very simple and not animal dispersed, do you have any new understanding from your adaptation studies that indicate how any climate change distribution changes could be tracked by dispersal of the plants as habitat patches move within respect to elevation or other geographic patterns?
Yeah, that's a great question. And I think I may have simplified how boring the dispersal mechanisms are, because when we go out to the field in the fall to plant our experiment for the next year, it's often really, really windy. And I've wondered for a long time how much, and this is when seeds are being dispersed. So sometimes the seeds get dispersed earlier and they just fall to the ground. But I do wonder how much the amount of wind in the fall may redistribute the seeds. And it's something that we're really keen, your question is, we're really keen to be able to answer your question, and I don't have a good answer at this point. What I really want to do is model the population in the future and assess how much gene flow would be necessary for populations to maintain positive population growth rates in the future in these new environments. But we don't yet have those data. We do have some sequence data from which we should be able to assess gene flow rates at a finer scale than what we currently have.
Great. And that was our last question for you for tonight, which it's just about 7 o'clock, so we do need to start wrapping up as well. So yeah, for those of you who might have missed the introduction, we do have a next webinar on January 21st, and you can also find Dr. Jill Anderson's talk on our website as well. And Jill, that was a really wonderful talk. Thank you so much for joining us for Science Cafe. It was a really interesting and engaging presentation, and I loved all your photos. So anyways, you guys should definitely check out Dr. Anderson's website. She's got some really great info on there. I've got her Twitter handle available for you as well. And then, like I said earlier, you should get a little survey for some feedback about our science cafes after this webinar. If you don't get that, feel free to email me, put my email here. And oh, my slide's a little messed up, but don't forget to follow us on Instagram, and definitely keep in touch and check out some of our future cafes. Thanks, everyone. Have a good night.
Thanks, Lila. That was great.
Yeah, that was so much fun to have you. Thank you for joining us. And yeah, hopefully we can have you again.